Hey everyone, this video is going to be all about the antibiotics, which is one way that we control bacterial growth. So let's go ahead and talk about what antibiotics are possibly going to target. Well, they are designed, well, they're not designed. Antibiotics occur naturally in the environment. Synthetic antimicrobials are made in a lab. So, but no matter whether it's a naturally produced or synthetic antimicrobial, they are designed to target key structures in the cell that are critical for the organism's survival, as well as key processes that are critical for the organism's survival. So we're going to do a lot of reviewing from lecture 1 or unit 1 content. So one of the first key structures that several antimicrobials target are the cell wall. And that is because if the structure of the cell wall is compromised, it can't withstand turgor pressure, which is the pressure from, fluids like water entering or exiting the cell. So we're kind of thinking back to osmosis. And so when they can't withstand turgor pressure, they become very fragile and lysis typically occurs, which is cell death. So let's talk about a few of the antimicrobials that are going to target the synthesis of the cell wall. So there are 4 different ones that you want to know, and you're going to want to know their name and their mechanism of action. That's what MOA stands for, and that's essentially how they work. So for 3 out of the 4, I'll have pictures to kind of go along with them, with the explanations. All right, so first up are the beta-lactams, and these antimicrobials have a very specific ring structure that is part of their overall structure, and that ring is called a beta-lactam. And so all of these antimicrobials have that unique ring structure called the beta-lactam. So the 2 beta-lactams that you need to know are penicillins and cephalosporins, and they have a similar mechanism of action, which is to bind to enzymes that are responsible for cross-linking peptidoglycan strands. So remember, the cell wall in bacteria is composed of peptidoglycan, and so the glycan are the sugar units, and peptido is for the proteins. And these are important for cross-linking, and you'll see this on the visual on the next slide. But essentially, beta-lactams prevent cross-linking amongst the glycan strands, which means it's preventing creating, especially for gram-positives, that thick peptidoglycan layer. Next up is bacitracin, and it prevents the addition of new NAG and NAM subunits. And so remember, peptidoglycan, the glycan part, are repeating units of molecules called NAG and NAM. And so because the cell wall is made outside of the cell, those individual subunits that make up the peptidoglycan have to be transported outside of the cell where they're then constructed, where the cell wall is then put together. And so bacitracin prevents those subunits from leaving the cell. Okay, so they can't make new peptidoglycan, and there are actually going to be holes in the wall where it hasn't been built, essentially. And so I remember this one because I think of BAC, of bacitracin, like get back. It prevents the units from actually leaving the cell. So I kind of imagine a little guard being like, nope, 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 get back. You cannot exit. All right. Next up is vancomycin, and vancomycin is going to also prevent the cross-linking of those glycan strands, but it does so a little bit differently than our beta-lactams. Vancomycin is actually going to bind itself to the glycan units, and therefore blocking the protein cross-links that would help make it nice and connected. And again, there'll be a visual of this in just a minute. And then we have tacobactin, which is going to target lipids in the cell wall, primarily teichoic acid. So think of which type of bacteria are most susceptible to this bacteria. Okay, so and I don't have a visual for this one, but it's just going to be targeting the lipids, which is going to compromise the integrity of the overall cell wall, again, making it more fragile and more susceptible to lysis. All right, that's the end of our antimicrobials that target cell wall synthesis.